Let's use this mind map to remember um, gluconeogenesis. There are substrates for gluconeogenesis like the lactates, glycerol, amino acids, the unique steps in gluconeogenesis, energy cost of gluconeogenesis, the summary of the Cori cycle, and gluconeogenesis, regulation of gluconeogenesis, the location of gluconeogenesis being the liver and kidney, non-muscle, and its gluconeogenesis connections like pyruvate in glucose out, lactate in glucose out, or alan- alanine in glucose and urea out, and also the ATP costs when it comes to gluconeogenesis, like 2 lactates, 6 ATPs, equivalent glucose, and also the primary signal when insulin turns off, glucagon turns on, acetyl CoA turns on, phosphorylation turns on in the liver. Yeah, so we try and then remember some of the source and substrates of gluconeogenesis and the unique steps and enzymes involved in the entire gluconeogenesis process or pathway. So remember that a number of tissues like the brain, size and muscle, red blood cells, kidney, medulla, testis, the lens, and cornea require continuous supply of glucose as a metabolic fuel. And during a prolonged fast, like 10 to 18 hours, the liver glycogen stores are depleted and glucose is formed from precursors such as lactate, pyruvate, glycerol from fatty acids, and alpha keto acids from proteins by gluconeogenesis. So the thing is, these precursors could be acquired from the glycolysis glycolytic pathway, the D um HAP, which is being converted to glycerol or back there. You can get glycerol being involved over there, and with the TCA cycle, amino acids, and with pyruvate could be converted to alanine and also to lactate. So that's where you get your precursors that are being utilized in gluconeogenesis. So the liver is a major site for this process where approximately 90% of gluconeogenesis occurs during an overnight fast, and during a prolonged fast, the kidneys may account for as much as 40% of glucose produced. And the capacity to synthesize glucose is essential for survival in the absence of glucose. When it comes to a substrate for gluconeogenesis, lactate is released into the blood from exercise and muscle. Um, that is taken up by the liver as part of the Cori cycle. So the lactate is reconverted to glucose in the liver and released back into the bloodstream. So with the Cori cycle, remember you have your muscle, you have your blood, and you have your liver. So glucose is being converted to pyruvate through glycolysis. ATP is being produced. NAD plus is utilized. NADH is being produced. All these happening in the cytosol. Pyruvate gets into the mitochondria. You have your TCA cycle. GTP and other products are being produced. GTP is being converted back to ATP. But then lactate is also being produced still in the muscle, and then it gets into the blood, and then it goes into the liver, and in the liver the lactate is converted back to pyruvate, and the pyruvate goes back into the cycle and is being into the mitochondria, is being reconverted back to glucose. So that's the Cori cycle, and it goes on and on and on. The glucose can go back to the muscle, and then the cycle goes on and on. So glycerol the glycerols, the fatty acids from esterified glycerol, are stored in adipose tissue and serve as the body's major fuel or storage reserve, and is released into glycerol is released into the blood following the hydrolysis of glycerols in adipose tissues, and is transported to the liver where it's phosphorylated by glycerol kinase. So the glycerol phosphates dehydrogenase can be converted or can convert the glycerol phosphate to dihydroxyacetone phosphate, that's a DHAP, which is an intermediate of glycolysis and gluconeogenesis. With amino acids, is derived from the hydrolysis of tissue proteins, and these proteins are the major source of glucose during prolonged fast. So catabolism of glucogenic amino acids yields pyruvate, or an intermediate of the citric acid cycle, or substrates for gluconeogenesis. And pyruvate and substances entering the citric acid cycle can form oxaloacetate, a precursor of PEP, which is the phosphoenol pyruvate, an intermediate of glycolysis or gluconeogenesis. So the unique steps of gluconeogenesis, there are 7 glycolytic steps are reversible and are used in gluconeogenesis, but 3 of the glycolytic reactions are irreversible, like the hexokinase step, the phosphofructokinase, and the pyruvate kinase steps, and are counted for by 4 alternative reactions during gluconeogenesis to favor glucose production. So the reactions of gluconeogenesis, the first enzyme, pyruvate carboxylase, is being implicated over here, and it carboxylates pyruvate to oxaloacetate in the mitochondria, and it requires a cofactor biotin. It is vitamin needed in the diet and ATP. And pyruvate carboxylase has an absolute requirement for acetyl-CoA. As an example of a Vmax control, that is enzyme rates increases with substrate amount. Low acetyl-CoA causes the enzyme to be inactive, and pyruvate can then be used to synthesize acetyl-CoA itself. With acetyl oxaloacetate is being reduced to malate, which can cross the cytosol. It can cross into the cytosol where it is deoxidized to oxaloacetate. So the second new enzyme, which is the phosphoenol pyruvate carboxykinase, as PEPCK, is located in the mitochondria in the cytosol in humans, and it decarboxylates and phosphorylates oxaloacetate to PEP as well. Phosphoenol pyruvate, and the reaction requires GTP. Okay. So GTP plus oxaloacetate, pyruvate, phosphoenol pyruvate, GDP, CO2 is being formed. The reactions of glycolysis are then reversible up to the level of fructose 1,6 bisphosphate, and the third new enzyme is now used since fructokinase is essentially irreversible. So this enzyme is fructose bisphosphatase, which interestingly is inhibited by AMP and fructose 2,6 bisphosphate. And finally, the fructose 6 phosphates form this equilibrated to fructose 6 phosphate, which is hydrolyzed to glucose via the fourth new enzyme, glucose 6 phosphatase, associated with the endoplasmic reticulum. So this bypasses the irreversible hexokinase reaction, allowing the formation of free glucose. And remember that the muscles lack this enzyme. With the clinical correlation, you can think of glucose phosphatase deficiency, which is responsible for von Gech disease, where type 1A or alpha glycogen storage disease, and is required for the final steps of gluconeogenesis and glycogen degradation in the liver. The premature infants and small neonates have a greater susceptibility for hypoglycemia, and due to a number of factors, the larger brain or body weight ratios and the brain's high demand for glucose, newborns have limited capacity for ketogenesis, that is transport of long-chain fatty acids into the liver, mitochondria, and low availability of ketone bodies as an alternative fuel for the brain. Glucose synthesis from lactates and alanine is limited because PEPCK is in low amounts during the first few hours after birth, and it takes several hours for PEPCK to be induced sufficiently to prevent hypoglycemia. And premature and low weight newborns have smaller store of glycogen, which makes them more dependent on gluconeogenesis than normal infants. And other disorders like glycogen storage disorder, hyperinsulinism, children of diabetic mothers, and stress that increases glucose consumption, like hypothermia, can cause exacerbated hypoglycemia. Intravenous glucose should be given until adequate feeding occurs. So, with the energy cost of gluconeogenesis, the energy from lactates you could think of 2 lactates to make 1 glucose. The Cori cycle just represents a net gain for efficiency of anaerobic glycolysis in the muscle by shifting the metabolic burden to the liver. But how is the ATP acquired in the hepatocytes? That one we've already talked about them. So lactate to pyruvate to phosphoenol pyruvate to glucose phosphate, and then to glucose in the liver. And the Cori cycle, like I said, is glucose in the muscle to lactate in the muscle to lactate in the blood, lactate in the liver, converted back to glucose, and glucose moves to the blood and moves back to the muscle, and then the cycle continues and on and on. So roughly about 6 ATPs are required to generate 1 molecule of glucose in the liver, and 2 ATPs are generated by anaerobic glycolysis in exercise muscles, and this cost of 4 moles of ATP cannot be sustained indefinitely. So during starvation, a triacyl glycerol hydrolysis in adipose tissue releases fatty acids into the blood, and then the ATP is needed in the liver for gluconeogenesis, and is provided largely by fatty acid oxidation. The other pathways of gluconeogenesis, there are a number of substrates other than lactates that can serve as gluconeogenic precursors, like alanine, pyruvate, and then glutamate, alpha-ketoglutarate, and CCA cycle, and several other amino acids feeding into the citric acid cycle are the major sources of glucose during fast. The other pathways, carbohydrates such as fructose and galactose, glycerol, and even short-chain fatty acids. Glycerol is released during the hydrolysis of triacyl glycerols in adipose tissues, and so galactose from glucose 6-phosphate, the major precursors of gluconeogenesis. Fructose at the glycerol dietary phosphate steps, glycerol also being produced, lactates from pyruvates, alanine from pyruvates, TCA cycle, you have your propionates and amino acids. So the regulation gluconeogenesis actually can occur in several ways: through mass action or non-covalent allosteric regulation. So in mass action, that is availability of substrates. For example, the direction of glycerol phosphate dehydrogenase is readily switched by NADH and 1,3 bis phosphoglycerate levels being high, which would ensure gluconeogenesis. The other major way of regulation of gluconeogenesis, like I said, the non-covalent allosteric regulation. A number of enzymes in the pathway are regulated by allosteric effectors, most often by changing substrates PM. And in general, the negative effectors of glycolysis are positive effectors of gluconeogenesis, and the regulation of the activity of PFK1 and F1 cis-base phosphatase is the most significant site for controlling the flux towards glucose oxidation or glucose synthesis. It is predominantly controlled by fructose 2,6 bisphosphate, which is a powerful negative allosteric effector of F1 6-bisphosphate bisphosphatase activity. A third one is covalent regulation, where phosphofructokinase PFK is indirectly regulated, and also pyruvate kinase for regulation of the liver by glucagon, which stimulates gluconeogenesis. Another point or another factor is energy enzyme amount. So it changes in the diet, that is high diet, high fat diet, high carbohydrates, or starvation can have major effects on the concentration of certain enzymes in these pathways. For example, the pyruvate, carboxylase, and PEPCK. Right, so that is it for the gluconeogenesis. So just a quick recap. Gluconeogenesis is the process by which non-carbohydrate molecules are converted to glucose, and the process occurs in the liver and kidneys. And during starvation and prolonged exercise, when blood sugar is low, the glycogen stores are depleted, and the formation of new glucose becomes the only means for maintaining constant blood sugar levels. And this pathway is triggered by glucagon and cortisol. So the gluconeogenesis pathway occurs mainly in the cytosol, but partially in the mitochondria. And with the non-carbohydrate molecules, you can think of lactate acids, which are converted back to pyruvate in the liver. That is a Cori cycle I talked about. And amino acids, for example, alanine, cysteine, glycine, serine, threonine, they are all converted to pyruvic acid or pyruvate. And these are glucogenic amino acids. For glycerol from fat, may be converted back to a glycerol phosphate or dihydroxyacetone phosphate, that's a DHAP, which may be used for glucose synthesis. So comparing gluconeogenesis and glycolysis, gluconeogenesis involves several enzymes of glycolysis, but it is not a reversal of glycolysis. Remember that. So there's 7 glycolytic reactions which are reversible and are used in the synthesis of glucose from lactate or pyruvate are being used over here, but 3 of the reactions are irreversible, remember, and the irreversible steps in the glycolysis are circumvented by 4 enzymes which are designated as the key enzymes of gluconeogenesis. So the formation gluconeogenesis, remember, there's a formation of glucose from glucose 6-phosphate by glucose 6-phosphatase, and the conversion of fructose 1,6-bisphosphate to glucose fructose 6-phosphate by fructose 1,6-bisphosphatase, and also synthesis of PEP from pyruvate requires 2 more enzymes: the pyruvate carboxylase, which converts pyruvate to oxaloacetate, and also PEP carboxykinase, which carboxylates and phosphorylase oxaloacetate. So with the pyruvate carboxylase reaction, the um, pyruvate in the cytoplasm enters the mitochondria, and then carboxylation of the pyruvate to oxaloacetate is catalyzed by the pyruvate carboxylase, and it needs the coenzyme biotin and ATP. And PP production in the cytosol requires the transport of oxaloacetate out of the mitochondria. However, there is no transporter for oxaloacetate in the inner mitochondrial membrane, so it's first reduced to malate by malate dehydrogenase, and the malate is then transported into the cytosol and re-oxidized by oxaloacetate by the malate dehydrogenase as NAD plus is reduced to NADH plus. And yeah, so you see the pathway, so like glycolysis from glucose phosphate to glucose to glucose phosphate, fructose phosphate, you realize step 1 is irreversible 4, and then the last step 2. So pyruvate is converted to oxaloacetate, which cannot get back to the cytosol but will have to be reconverted back to malate before it gets out, and it's being reconverted back to oxaloacetate. And then the whole process goes on from the phosphoenolpyruvate to the 2 phosphoglycerates back to 3 phosphoglycerates, 1 3-base phosphoglycerate to glyceraldehyde, 3 phosphates to fructose 1,6-base phosphate, back to fructose 6-phosphate, back to glucose 6-phosphate, and finally to glucose. So the phosphoenolpyruvate carboxykinase in the cytoplasm, this enzyme converts oxaloacetate to phosphoenolpyruvate by removing a molecule of carbon dioxide, and GTP or ITP donates the phosphate, and the net effect of these 2 reactions is the conversion of pyruvate to phosphoenolpyruvate, and this circumvents the irreversible step in glycolysis catalyzed by pyruvate kinase, which is step 9 of glycolysis. So fructose 1,6-base phosphate dephosphorylation, like I talked about, the hydrolysis of fructose 1,6-base phosphate by fructose 1,6-base phosphatase is found in the liver and the kidneys and bypasses the irreversible phosphofructokinase 1 reaction of glycolysis and provides an energetically favorable pathway for the formation of fructose 6-phosphate. And then so from here, from what is being utilized over here, inorganic phosphate is being moved out, citrates AMP F2 6P, they are all being removed from here. So with the reaction, the glucose 6-phosphatase reaction, glucose 6-phosphate is hydrolyzed to free glucose by glucose 6-phosphatase, and it's active in the liver. This particular enzyme, the glucose 6-phosphatase, is active in the liver and is present in the kidney and intestinal mucosa to a lesser extent, but it's absent in muscles. So the glucose 6-phosphate plus H2, presence of glucose 6-phosphatase, you get glucose and inorganic phosphate. Okay, so the cycle is very simple. So pyruvate, 2 pyruvate molecules is being converted to oxaloacetate via pyruvate carboxylase, utilizing 2 ATPs and HCO2 CO3, so, and then releasing ADP as a byproduct, and the enzyme PEP carboxykinase converts oxaloacetate to PEP via utilizing two GTPs and then forming two GDP molecules and two CO2 molecules as byproducts, and the PEP is being converted back to pyrophosphoglycerate, and that is being converted back to 1,3-bisphosphoglycerate by phosphoglycerate kinase utilizing two ATPs over here. So from the two molecules of 1,3-bisphosphoglycerate, it utilizes NADH and one hydrogen ion, which will release NAD plus, and then there's a formation of fructose 1,6-bisphosphate, which the enzyme this 1,6-bisphosphatase converts it to fructose 6-phosphate, releasing inorganic phosphate as a byproduct, and then the fructose 6-phosphate is being converted to glucose 6-phosphate, and this is being converted back to glucose by glucose 6-phosphatase, where there's a release of one inorganic phosphate. And a recap of the Cori cycle: in the muscle, glucose converted to pyruvate and to lactate. The lactate moves into the blood, and the blood from lactate from the blood moves into the liver. The liver is converted back to pyruvate back to glucose via gluconeogenesis, and then the glucose goes back to the blood and back to the liver. Glycolysis takes place, lactate, and the cycle goes on and on. So the exchange of metabolites between the liver and the skeletal muscle is what we call the Cori cycle. And during exercise, blood lactate level increases considerably, and the lactate then reaches the liver where it's oxidized to pyruvate. It's then taken up through gluconeogenesis pathway and becomes glucose, and then enters into the blood and is taken to the muscle. So the significance of the Cori cycle is that muscles cannot form glucose by gluconeogenesis process because glucose 6-phosphatase is absent. But unlike the liver, the muscle cannot supply glucose to other organs in spite of having glycogen. So that's it for gluconeogenesis. We'll continue with PPPs.